Hello everyone, how are you today? I hope you're guys doing well. And today we're gonna discuss about one of the interesting topic in biology, which is a virus. Let's get started. Here's the outline for our discussion today. There will be a preliminary history of the discovery of scientists about the viruses, the characteristics of the virus body structure, virus reproduction or replication, and the last is the role of the virus in our daily life. Let's go to our first topic, which is the preliminary. The study of viruses is called virology. Virus in Latin is virion, which means poison. Viruses are acellular organisms that do not have organelles. And this is the history of virus discovery. The first guy was Edward Jenner in 1749 until 1823. He was the first scientist to find a vaccine to prevent smallpox or variola. Smallpox is caused by a virus. The second guy was Louis Pasteur. He was a French scientist who managed to find the center of infectious disease found in the brain and spinal cord. In addition, he was also able to transfer the infection to laboratory animals in the laboratory. The next is Adolf Meyer, Germany guy, in 1893, and he stated that the tobacco spots were yellow spots caused by organisms smaller than bacteria. One of the proofs, spraying the sap of diseased plants into healthy plants turned out to be infected. The fourth guy was Dmitry Ivanovsky, Russian guy, in 1893. He is a botanist who discovered tobacco plants exposed to mosaic disease, then filtered through very fine pores that are still able to infect tobacco leaves. And the fifth guy was M.V. Beijerinck, Dutch guy, at 1897. He stated that tobacco disease contains a living body of cell that called a virus. And this timeline shows you the history of virus discovery. The first is by Wendell Stanley in the United States on 1935. The second is Loeffler and Forts at 1897. Scientists from Germany. Viruses that attack animals, namely nail and mouth disease in livestock. And the third was rat at 1900. He found that the virus caused dengue fever in humans. The disease is caused by Aedes aegypti mosquitoes. The next is Laudrow and Stuart Harris on 1933 until 1936, which discovered influenza viruses. And the fifth is Jonas Salk at 1953, which discovered a vaccine against polio. Mayer's experiment on virus research. The first is he found a tobacco disease plant, and the leaves are mashed, and then he make a leaf extract, and the leaf extract from the tobacco disease plant, he sprayed that to the healthy tobacco plant, and after that he observed the healthy plant become ill. The next is Dmitry Ivanovsky experiment on virus research. The first is leaf of tobacco, once disease mashed up, and then he filtered the leaf and make it an extract of tobacco disease. After that, the filtered leaf of tobacco sprayed to the healthy plant, and the same result with the previous experiment. The tobacco plants which is healthy become ill. Then, let's figure out what is a virus. Basically, viruses are particles that obligate parasite in cells or living things. Viruses are acellular or not a cell. They are very tiny size, and inside the host cell, the virus shows the characteristic of living things, while outside the cell shows the characteristic of non-living things. Here is the characteristics of viruses. Viruses have the following characteristic. The virus body size average is around 0.02 until 0.3 microns, or also the smallest virus is the poliovirus. The body structure of virus is consist by two things. The first is genetic material. It can be DNA, deoxyribonucleic acid, or RNA, ribonucleic acid surrounding the layer, and capsid, which is a membrane composed of protein units called capsomers. Virus has diverse body shapes. There are longitudinally rectangular or flamen, T-shaped, and stem, or cylindrical and round viruses. The virus has two phases, namely inside the host cell or outside the host cell. Host cell, namely living cells infected by parasites. Viruses also can be crystallized, but if they are in cells or living tissues, they are capable to replicate themselves, and they can be called transitional creatures. And in general, viruses do not have plasma membranes, cytoplasm, and ribosomes, so they are not yet referred to as a cell. This is some comparison from the virus size. For the picture A, you can see this is the virus particles in green dots. And for B, this is a bacterial cell. And the last picture, C, is the eukaryotic cell. You can see that the virus particle is very small. It's around 0.2 micrometer, which is 1/5 size from the bacterial cell or eukaryotic cell. The next is examples of virus characteristics. The first picture shows you the round virus. This is an influenza virus. And the second picture is oval virus, which is a rabies virus for this example. And the third picture is a stem virus, which we previously said about tobacco mosaic virus, or TMV in abbreviation. And the fourth is something quite complex, which is a polyhydric virus. This is an adenovirus which causes fever. And the last is T-shaped virus, which you may see a lot. This is a T-shaped virus, and the example is bacteriophage or phage. The virus classification above is based on the body shape of the virus itself. Our next discussion is about structure and function of parts in the virus particles or virions. The particle itself called virions, and it's divided into two, which is outside part and the inside part. The outside part consists of two, the capsid, which is protein, and the protein envelope, which is a nucleic acid wrapper. Inside part is divided into two, which is nucleic acid or genetic materials. It can be DNA or RNA, and various proteins or enzymes. Some of the structure parts of the virus. 1. Capsid are arranged by protein. A capsid forming unit is called a capsomer. And the core material consists of nucleic acids, which consists of only DNA or only RNA. Nucleic acid function to control the replication or reproduction activity of virus. And the head is composed of nucleocapsids in the form of polyhedral or multifaceted, which is inside there are nucleic acids and outside are composed of capsid. The last is in some virus, the outer portion of the capsid is found to have a viral envelope or a membrane that surrounds the capsid originating from the host membrane. This sheet is composed of phospholipids and proteins from the host cells as well as proteins and glycoproteins from virus. The viral envelope functions to help infect host cells and carry several enzyme molecules. This picture is showing you about the virus body structure. This is a veiled virus. As you can see, the outer side is the virus receptors, and then there are virus capsules, which is shaped like a circle here, and inside we can see the protein sheet or capsid along the genetic material inside, which could be DNA or RNA. This is the example of complex virus, what I said previously before. I said that this is an example of bacteriophage. Consists of capsid, which inside there's DNA and then tail sheet and tail fibers. And the next picture is showing you the helical virus and Ebola virus. For the helical virus, you can see there is a capsid and capsomer, also nucleic acid inside the capsomer. This is a virus body structure of the Nagg virus. Other scientists consider that viruses are living things. The reason is the virus can show the characteristic of life, namely reproduction, and it's carried out in the organism cells. So it can be concluded that the virus is not a cell because it does not have cell parts. For example, the plasma membrane, cytoplasm, and nucleus. And the next is we're gonna discuss about the mechanism of virus replication. Viruses show the characteristics of life, which is reproduction inside the cell of organism. Cells in which organisms are called host cell. Then the virus can live parasitically. The purpose of replication itself is to multiply the virus particles. The virus replication occurs only when the virus is in the cells of living things. In general, it occurs through 5 stages. The first is absorption, the second is penetration, the third is synthesis, the fourth is maturation or assembly, and the fifth is lysis. Divided into 2 cycles, namely lytic cycle and lysogenic cycle. Here, we will talk about both of the cycle one by one. As you can see, this is the cycle of lytic cycle and lysogenic cycle. Lytic cycle is depicted in the left side and the lysogenic cycle is depicted on the right side. The lytic cycle has 5 stages. The first is absorption stage, or you can see in the picture is attachment. And the second is penetration or entry of phage DNA and degrade the host DNA. The third is the synthesis of viral genomes and proteins. And then the maturation or assembly. And the last step is release or lysis of the virulent particle itself. For lysogenic cycle, this cycle is named lysogenic because the host cell at the end of the cycle does not experience damage or death. Host cells can survive and not be damaged at the end of the cycle because due to the host cell have resistance to virus attacks that called virulence. Lysogenic cycle is slightly different with the lytic cycle. For lysogenic cycle, it's only occur for 4 stages, which are the absorption, penetrating or injection, the merging, because at this phase, DNA is inserted into the bacterial DNA so that the bacterial DNA contains a virus genetic material. Virus DNA that has inserted into bacterial DNA cannot be active to take control of metabolism from bacterial DNA because bacteria have virulence. And the virus DNA that attach to bacterial and inactive DNA is called prophage. And the last stage is cleavage. In this phase, prophage will be in the body of bacteria as long as the bacteria still has virulence. When the bacterial cell undergoes division, the DNA of the virus is also copied so that 2 bacterial cells are formed, each of which contains a prophage. Bacterial cell division can be repeated in several generations, and the profile will also be divided into several generations. Also, the profile will be divided into several generations. And this table shows you the difference between lytic and lysogenic cycle. For the variable bacteria or host cell initial condition, the lytic cycle is non-virulent, and for the lysogenic cycle, the host is virulent. For number of stages, lytic cycle have 5 stages and the lysogenic has only 4 stages. For the cycle continuation, the lytic cycle is stopped because the host cell is damaged or undergo lysis. But for lysogenic cycle, It can be continued with the lytic cycle if the virulence of the bacteria or host cell is lost. And the final condition is bacteria. For the lytic cycle is death, and for lysogenic cycle is do not undergo lysis or not death. The next topic is the virus classification. It's divided into four, which is the first is the bacterial virus or bacteriophage. The second is protist virus. The third is the plant virus, and the last is animal or human virus. And now we are in the last topic of our discussion. This is the role of the virus in daily life. These are the harmful effects of the virus. Most of them cause disease in humans. The first is influenza or flu. It attacks the respiratory tracts. The symptoms of this disease include the runny nose, cough, and dizziness. The second is smallpox. It's caused by the herpes virus, varicella virus. And the third is the measles. It's caused by the paramyxovirus virus. The symptoms of this disease such as the high fever, cough, and pain throughout the body. The fourth is hepatitis or swelling of the liver. There are three types of hepatitis virus, namely A, B, and C. The most dangerous is the B virus. The symptoms are fever, nausea, and vomiting, and jaundice symptoms appear. The fifth is polio. In general, attacks children. The symptoms are fever, headache, not feeling well, drowsiness, sore throat, nausea, and vomiting. And the last is herpes. It attacks the mucous membranes in the mouth, genitals, eye, and skin. Another harmful virus are rabies or crazy dog, AIDS or acquired immune deficiency syndrome, which caused by HIV, and Ebola, virus DHV, virus trachom, and the poliomyelitis. And this is the disease in animals caused by virus. NCD or Newcastle disease, it attacks the nerves of the poultry. For example, chickens and ducks are often called tetelo or adshore. The next is RSV or cancer in chickens, and there are FMD, livestock, rabies, and smallpox. However, the virus has some favorable effects such as virus can be used in the field of genetic engineering or gene mutation factors as well as research in medicine. The virus is also used to produce interferon functions to prevent the replication of the virus in host cell. Interferon is a small protein produced by normal cells in response to viral infection, or it can be said as like an antigen. The last is the viruses are used to make vaccine. Vaccines are pathogenic microorganisms that have been attenuated so that the pathogenicity of the cause of the disease is lost, but the properties of antibody generating antigenity remain. And another information about the virus, as we know, we are now in a pandemic situation and it's caused by the coronavirus. So let's talk about this in a little scope. Coronavirus disease or COVID-19 is an infectious disease caused by a newly discovered coronavirus. Most of the people infected with COVID-19 virus will experience mild to moderate respiratory illness and recover without requiring special treatment. Older people and those with underlying medical problems like cardiovascular disease, diabetes, chronic respiratory disease, and cancer are more likely to develop serious illness than the healthy one. Because this is an infectious disease, then the best way to prevent and slow down the transmission of COVID-19 virus, the disease is caused and how it spreads. And then protect yourself and others from infection by washing your hands using an alcohol-based rub frequently and not touching your face. And at this time, there are no specific vaccine or treatments for COVID-19. However, there are many ongoing clinical trials evaluating potential treatments. WHO will continue to provide updated information as soon as clinical findings become available. Therefore, I hope you can keep yourself safe and stay safe, everyone. That's all for our discussion today. Hope you understand about virus and how to overcome with the virus. See you.